Okay. So can I introduce Victoria? So Victoria is a postdoc in our lab, and she's rapidly growing as a kind of a scientist, professional scientist, researching into Aniridia. So Victoria?
Hi everybody. Yes, so I'm going to tell you about my project, which I'm working on at the moment, which is to try and develop a human disease model for Aniridia-related keratopathy. So I'll just tell you a bit about how I got where I am. I started my PhD in Norwich at the University of East Anglia, where I worked on the cell biology of retinal detachment. I became very interested in ocular disorders and the cell biology behind these disorders. So I continued, when I moved to London, I got my first post working at UCL, and I was working on wound healing following glaucoma surgery. So as an academic scientist, you don't have a permanent position. You tend to have 2 to 3 years of funding for a particular project, and then you have to either get more funding, which is usually quite difficult, or you go on and find another post somewhere else. So that's where I am now. Luckily, I met Julie Daniels in the team, and I'm now working on my project to do with Aniridia-related keratopathy, which I'm going to call ARC from now on. So I'm sure you guys know a lot about ARC, but I just want to highlight some of the cellular conditions associated. So in a healthy cornea, you have the outermost layer, which is the epithelium, which is made up of a number of layers of epithelial cells, generally about 5 or 6. The layer underneath this is the stroma, which is the majority of the thickness of the cornea, and the stroma is made up mostly of collagen, which is a fibrous protein. And this collagen is organized really highly by specialized cells in the stroma, and these cells are called keratopathies. Underneath the stroma is the endothelium. But I mostly work on the stroma and the epithelium, so the top 2 layers of the cornea. Some of the problems associated with ARC are an abnormal or a weak epithelium, and there's also impaired wound healing and impaired wound healing response. Epithelial stem cell failure, which contributes towards the clouding of the cornea due to an infiltration of conjunctival cells and blood vessels. So in the lab, we can grow all 3 types of corneal cells, and we do this routinely. And we can do this by growing them from human donor tissue. So we have a system where we can grow human corneal cells in the lab. And with these cells, we can then build a tissue-engineered cornea, which is used often in surgery that Alex is talking about next. But also we can use it as a model. When I say model, I just mean a system in which we can try and understand what the cells are doing, how they're behaving, and ultimately, hopefully try and figure out what's going wrong in the disease process. So we can build this tissue-engineered cornea by using a liquid collagen. Like I said, collagen is the protein that makes up the stroma. And we can use this as a liquid, and we can mix it with the stromal cells that we've grown. And it's a bit like jelly. We can then leave it, and it'll set into a sort of solid, sort of wobbly thing. And then once it's set, we can then put the epithelial cells on top and leave them to grow. And over a couple of weeks growing, they'll start to multi-layer, just like you see in the human cornea. So the aim of my project is to try and make this tissue-engineered cornea, but make it aniridic. So I want to try and make faulty epithelial cells and put these cells onto the tissue-engineered cornea and see what happens basically to the cells. So a bit of science, if you're ready. So I needed to find out if the epithelial cells that I grow express Pax6. Sorry, I should have said, I'm going to make the faulty epithelial cells by reducing Pax6 expression in the cells. So one of the methods that we can use to do this is called immunofluorescence. So we can use this method to find out if we have Pax6 in our cells. And the reason it's called immunofluorescence, it's immuno because we use antibodies. antibodies that are specifically targeted to proteins of interest. So I can buy an antibody for a Pax6 protein, and that will bind to the Pax6 protein in the cells. And the fluorescence part is we add secondary antibodies which are tagged to a fluorescent fluorophore. So when we add these onto the cells, they bind to the primary antibodies, and then we can visualize the Pax6 inside the cell. So when I did this method, I found Pax6 was expressed. So if there was no Pax6 expression, you would just see nothing when you look down the microscope. But when I look down, I see nice green areas where Pax6 is expressed. You can also stain the nucleus of these cells, so you can find out where the protein is located. So you have a kind of reference point. So the nucleus we can stain with specific dyes. And you can also stain the cell skeleton, so much like as cells have the skeleton that holds the cell together and in place. So I found that I do have Pax6 expressed, and it is in the nucleus, which is where it should be expressed.
What do you mean by expressed?
Sorry, it's there. It's,
right.
Yeah, sorry.
The protein is made from the gene. It isn't in every cell. It's made in certain cells.
Yeah, so every single cell in your body has the genetic information for the entire genome. But each individual cell, say from a liver cell, you get different information in the cell which tells it to use that genetic information to make a protein out of it. So obviously in the nucleus of the cell are the chromosomes, which are made up of the DNA. The DNA carries the genetic information, and the process by which this genetic information is translated into a protein can be prevented by using certain compounds, certain things that we give to the cells. And one of these things is called SiRNA. It doesn't really matter what it's called, but this basically stops. You can have specific siRNAs, just like you can have specific antibodies that target different proteins or different mechanisms to make a protein. So I can use an siRNA that will stop Pax6 from going from the genetic information to making the protein. So no protein will be made of Pax6. And when I add this to my cells, I do get a reduction. And then I look at whether Pax6 is there or not. I get a reduction in the level of Pax6. So compared to control healthy cells, I see about an 80% reduction in Pax6 when I use this compound on the cells. So there is still a bit there, but it's enough of a difference between the control cells and the Pax6 siRNA cells to give us something to work with to see if there's an effect in these cells. So what effect does it have? In a healthy epithelium, the cells stick to each other. They kind of anchor to each other and to the top of the stroma, and this is by specific proteins as well. So they kind of stick to each other really tightly, and when we grow these cells in the lab, we can see a similar thing happening where the cells grow in groups together, and they don't tend to kind of distance off from those groups. They grow in the groups and they stay together. But when I look at my cells where I've reduced the Pax6 expression, if you can see this, there's quite a lot of areas where cells have kind of drifted off away from the group and are just kind of on their own, not really sticking together. So this is a good indication that they are losing the kind of structural integrity that they would have when they're in the corneal epithelium. And I can further investigate this by looking at different proteins to see, to confirm the findings here. Another experiment I did was to look at how fast they grow, because there's been reports by the scientists where there's a difference in the rate of growth when Pax6 is changed. So in my cells that have reduced Pax6 level, they grow at a much faster rate than my healthy cells. So this could also be contributing to the abnormal epithelium or the weakened epithelium. which is great because now I've seen that I can reduce the Pax6 level in the cell, and I can see that there are differences between control healthy cells and cells that have reduced Pax6. So then, for my future work, I'd like to put these unhealthy Pax6 reduced cells onto my tissue-engineered cornea, and then see if they multi-layer, see how they change, how they interact with other cells, whether the stromal cells make a difference, whether they're or whether they're not there. Just lots of things we can look at to find out have a better idea of what's going on with the Pax6 deficient cells. While I've got your attention, I just want to draw on a few funding points. Like Alex mentioned, we need to advocate eye research, really. We need to get out there that it is important to do research in ocular disease. Quite interestingly, I found out recently that we spend £30 million a year on eye research in the UK, but eye healthcare problems cost the UK approximately £8 billion, both with direct and indirect costs. And also that over 50% of sight loss could be avoided. So I think it's really important to get across to the general public and the government, hopefully, that we need to maybe increase the amount of money that we spend on eye research to decrease the other numbers, decrease the cost, as well as all the sight loss that can be avoided. We'd rather not have the 50% there. So, and also by 2050, it's estimated that 4 million people will be affected by visual impairments. It's meant to double by 2050. So, yeah, if you hear of anybody wondering anything about science, just tell them it's great, and we need more money. I'd really like to thank as well, speaking of funding, the Shine the Light on Aniridia project. So Ellie Chapel goes to a lot of effort to raise money for research into Aniridia. So that's great. I mean, that helps a lot towards the project that I'm doing. Also like to thank the special trustees of Moorfields Eye Hospital who have given me additional funding for some of the expensive things that I use in the lab, and also the Cells for Sight team. And thank you very much for listening. Are there any questions? Hi. Okay. Yeah. I think Alex might be better to answer that. So Aniridia related to keratopathy is on the cornea on the front, and the cataracts is in the lens of the eye. Oh, I see. Oh, wow, okay.
So obviously that makes things a little bit more difficult.
It's not really silly. It's like you've got Aniridia at the back, pieces of anomaly, but the only way I can describe it, but it is a piece of anomaly, but apparently can't have both. I mean, they won't do genetics, so we have no idea where it's gone wrong.
Yeah. I think I hope you're being looked after by someone who has an interest in either the anomaly and the law or right. I mean, I think you're in the best possible hand, and very often the thing about the NHS is there are very good things in getting a complex and finding and navigating the system.
Hold the piece of fact you've got.
Absolutely.
So we're basically, obviously, you haven't met anyone in like horror.
We know it's also, if this is a problem with the epithelial cornea, you've got a slight mark on it now, and cells go into sort themselves out. That's exactly explained now.
Well, in my next talk, I'll show you what we can do about that. Theoretically, these cell therapies that we're developing could be applied to children. We could use them to children
around and make sure we're